Hello, and thanks for joining us on Journal Club. My name is Alex, and we're going to be jumping into a pretty interesting publication today. I will first maybe pass it off to Jordan, and then we can do a round of introductions.
Yeah, thanks, Alex. So today we're going to be talking about an interesting paper that's related to solving some of the challenges with scaling cellular agriculture, primarily on the media side of things. I've got my notes here. It's titled New Glucose-Abstaining Chlorella Algae Doubles Mammalian Cell Culture Longevity, Boosts Performance, and Drops Serum Needs, Enabling Scaled Applications. The first author is Melanie Oey. I hope that's how you pronounce her name. And it's from the University of Queensland in Australia. The background is that cell cultures are limited due to high serum costs, frequent media changes, and waste accumulation, particularly when you scale up from flask to something like a bioreactor. So they're exploring symbiotic relationships that might be able to extend the longevity or the useful life of the media in these systems. They broke it down into a few main objectives. Number one, to be able to grow algae and at the temperatures that for mammalian culture at 37C, and they want to do so while avoiding competition for glucose, which is a big problem for these symbiotic systems. They had a couple of key claims that they outlined. For example, they identified the BDH-1 strain of algae to be a potential ideal strain for this application, given that they found that it did not seem to consume glucose from the mammalian cells. They also found that it improved C2C12 viability by up to 2.6x. And then it reduced FBS requirements by 50%. It lowered waste products such as lactate. These are all pretty substantial claims, and can't wait to really get into it with you guys.
Great, yeah. And before we kind of dig into the specifics of the publication, actually, I have a couple questions that came up right off the bat. But as being the first journal club, let's now go and kind of do a round of introductions. And so maybe we can start with Max.
Sure, happy to be here. I'm Max. I did my master's in Dr. David Kaplan's lab. I did a thesis investigating plant tissue culture applications as a scaffold like structure for cultivated meat. And I graduated last summer, actually, basically exactly a year ago.
Not a year ago to the day, is it?
I defended my thesis July 11th ish or something like that, so almost.
Okay, awesome. Rebecca?
Hi, I am Rebecca Lee. I have about a decade's worth of experience working in high content screening. So, mostly in early stage drug discovery, but going before that, Jordan actually were working together as biomedical engineers in undergrad.
Very cool. Jordan?
Sure. My name is Jordan Jones. I've been involved in the cellular agriculture space for about 7 years now. I actually started out researching cardiac muscle at Worcester Polytechnic Institute. I got my PhD in biomedical engineering from WPI, and then later worked with Dr. David Kaplan at Tufts University. And I've been continuing my research on cultivated meat structured products like steak and whole cuts of chicken breast, things like that, ever since.
My name is Alex Tarazi. I'm the host of a podcast by the name of the Future Food Show, and I have been hosting conferences about cultured meat since 2018. So now we're working on our 8th year, which is crazy to think about. I advise for different nonprofit and for-profit cultured meat companies, and I work with Jordan at Jones Cell Ag, which is a consultancy that works with cell ag and biotech companies. Now, going back to the article, and thanks for the overview, you know, maybe I'll start off with a question that, for those of you that are doing active research in cell culture or adjacent types of things, is there anything like this? Is this something you'd be like, oh, wow, I hope this could work for my research?
So, I've done work in organoids before, and it is a growing field of interest, I know, in pharmaceutical research. And being able to see better sustained organoids, especially with co-culture and with reduced media needs, because steroids can be quite delicate to do media exchanges on, is actually quite an interesting usage of this. But what I want to let the conversation see if anyone else has other inputs.
Sure, I can chime in. I'm not sure that there's much of an application for this at lab scale, but I do see some potential for this as you get larger and larger, scaling up to things like bioreactors, where you're talking huge volumes of cell culture media where the cost becomes really prohibitive. This can be anywhere from, I suppose, even at lab scale, when you move up to things like cell bags, for example, might be the first place where this type of research could be useful for recycling the media or even prolonging the media's use. But really, I see it as an enabler for more commercial applications in cell culture.
So I didn't expect us to my tangent so quickly, but I can bring us straight to my tangent then. I like how I think the idea of being able to use it at scale is really cool, but the idea I immediately thought of seeing the reduction in FPS usage was I know that every once in a while Jordan ends up chatting with NASA about bringing a cell cultured meat into space. But our conversations always cycle around the issue of media needs because of the weight of FPS and not knowing the media additions needed to safely and continuously culture meat in space. So the usage of macroalgae, my mistake, that's kelp, microalgae actually would probably enable this technology in space. One other detail I want to add, because I do have a little bit of an interest in marine science, is that one thing about some species of microalgae that makes it even more enticing is that it can survive desiccation. So you can fully desiccate the microalgae of certain species. So this was very clearly a lot of research went into finding this species. But desiccation is possible and thus even making it more viable for enabling space faring meat, cell cultured meat. And that is really what I saw as long in the future, maybe, but like I think was the most interesting possible application for this.
Oh, that's really cool.
Yeah.
Thanks for that.
The idea is that you would need such a small amount to take it up there, and then the algae
You
will
can
continuously.
expand it, and then you would be able to use that to reduce your FPS needs in space.
And also
Hmm.
lower the amount of materials weight that you need to take up
Yep.
there. Yeah,
Yep.
cool.
All of the
Yeah,
above.
yeah.
Because FPS, like you do, as the article mentions, that they do know some of the supplements that you can use for FPS. And to a certain extent, FPS can be powder desiccated and brought into space. But then it's like me and Jordan have gotten into this previously about the complications of creating standardized media that you mix up from powder. But like that could significantly reduce that hurdle of getting the amount of media you need in space in order to then later do cell cultured meat.
I'd love to get back into the dynamics of powdered media in a second, but one question I had, and Jordan, you were saying that you'd apply this more at a later stage, but wouldn't you also want to do testing early on to see how it reacts with kind of other things, or to your research, Max, like how it might react with the scaffolds?
Do you want to share your piece for a second?
Yeah, like how it would react with the scaffolds. So with the scaffold, I'd be interested. Do they talk about like the 3D structure? Do they set up actual experiments in the paper for the 3D structure of like perhaps chicken breast or like, simulations for that? Because I'd be interested to see kind of with the proliferation in space, if you set up like just kind of a seed of the algae, if you bring it to space and you have like these molds, that would be an interesting way to create the 3D structure and the hybrid scaffold with the cells that could then attach in space. DIY, you got the cells, you got the algae, bring it up, and then kind of grow them out there. I'd be interested to see the efficiency of the binding and if there's ways to increase that. And perhaps because it can grow in these, it can continue to grow up there, it can create that kind of not directly the marbling effect, but it could create like a unique structure that would be very interesting.
Yeah, just to help everyone visualize like what kind of experimental setup they had in this paper. They were using trans wells. So if you imagine a tissue culture weld plate, right? Let's just use a 6 well plate for example. You have an array, a 2 by 3 array of these wells on this plate. And at the bottom of each of the wells, you have your mammalian cells growing. So these are your C2C12s, which are mouse myoblasts typically used for any type of research about related to skeletal muscle. But the transwell part of it is essentially a basket that you put into the. So just imagine a hole, a well with a basket in it. The basket allows the algae to grow in the same media because that the bottom of that well is permeable, right? So the media can come through the bottom. So they're sharing the same environment, but they're distinctly isolated in a way. So they're sharing the same environment, the same media, and the concentration of different compounds in the media are free to cross, but the cells themselves are separated. So in this case, Max, it wouldn't necessarily be that the algae is contributing anything to the structure per se, but what they might do is they might provide an environment that might be more conducive to some of your calluses that you're growing that would contribute to the structure. Rebecca?
So I actually want to jump off what Max was saying, and I to point out the one weakness I did find in the article. I had some concerns. They didn't do too much like cellular staining work. I don't think at all. They only did bright field work inside the paper, and I was curious about whether or not there were noticeable morphological changes with or without the microalgae, which could speak to changes happening in the cells that we haven't really determined as of yet. Some things to look at would be whether or not you can run standard assays with these cells and get the same results. So there are questions, like there were no like functional testing of the cells to see how this cold culture is changing their behavior. So I definitely think it's very valid to wonder about structure and also all the other constituent processes happening inside the cells.
Yeah, I definitely agree on that part, particularly when it comes to differentiation, right? You have,
Hmm.
they use C2C12s, which I don't particularly as a model when it comes to
Yeah, that was an
cultivated
interesting
meat,
choice using the
right?
mouse model.
I don't particularly like it as a model, but it's still, just from a scientific point of view, it's still a valid one, right? Cell line. And those cells are capable of differentiating into mature myotubes. But we don't get to see any of that, right? They don't at any point in the paper differentiate the cells into
Hmm.
myotubes. And to your point, I would like to see how the cells differentiate on a more detailed level. Are they able to form sarcomeres? Are they expressing myosin heavy chain? Things like that. And it's just something I think would really add a lot of value to the study. Also, another point, one of my initial thoughts was that they used both for the algae, they used chlorophyll concentration, or rather the color of the chlorophyll, I believe, to quantify like how much algae is there. And in a way, I suppose that kind of works. But it doesn't tell us a lot about the viability of the algae, right? Just because it's there doesn't mean it's very healthy. It also doesn't mean that it's being very productive on the photosynthesis side of things. So seeing an assay that's more relevant to viability would be great. And also something that is closely tied to photosynthesis, such as DPIP. I only know this exists because if you want me to say the full thing,
Do it. I
I'll
want to
give
make
it a
fun of
try.
you.
I matter of fact, I'm just not gonna do that. One, I can't find it.
Doesn't want to open
I,
himself up to.
I don't want to open myself up to it. But the only reason I know about being able to quantify photosynthesis is actually because this study is very similar to one that my friend Rick Dyden did at Boston College. So he actually published a paper about heat resistant chlorella and how they could be used to recycle cell culture media. It was a fairly similar idea, but very different. And throughout that study, he used DPIP to quantify like how much the algae is photosynthesizing, and that's I think a very good measure of how well the algae is doing. But
I'm
even in terms, oh, sorry, go ahead.
no, you finish.
Even in terms of viability, both on the algae side and the cell side for the C2C12s, they use resizerin, which is
Yeah,
it's a measure of metabolic activity, which don't get me wrong, I think there's a lot of good data there, but it doesn't tell me a lot about their health, especially when it comes to an experiment which is going to directly impact the metabolism of the cells, right? The algae is playing a really big role in altering the metabolism of the mammalian cell culture. That's the point, right? They're supposed to help it, right? Help keep it stable. But my point is that it wouldn't be out of the question to see metabolic activity decrease. Metabolic activity decreasing doesn't necessarily indicate less viability. It just means the cells are moving slower. So My point is that the use of resizurin is not bad, it's just incomplete. If we included another assay, such as a cyquant assay to quantify DNA, it's going to be standard in every cell, be it live or dead, and couple that with your metabolic activity data, I think it would provide a much more complete picture of how the cells are doing. So that's it.
It also makes me think of what's the next phase or next step in their research, like what's going to happen next and who's going to continue that. I think that would be interesting. Also, kind of how much they'll focus on.
That is something I
Yeah,
would really like to see in any future research from them.
that actually brings me to my final point or note. The research is that though I think this helps with scalability from the point of view that it may be able to increase the longevity of the culture media, I'm not exactly sure. I think it still has some challenges on the scalability side. And that's because if you were to implement this in a large scale bioreactor, for example, I've worked in the pharmaceutical industry for a little bit, so I know I've worked with 1,000 and 6,000 liter bioreactors. And when you start to get to those scales, if you're doing like algae and you really start proliferating the algae, the optical clarity of that culture is going to decrease as the algae proliferates. I'm not an expert on photobioreactors or anything because that's what you would need for this. But as the clarity decreases as the algae proliferate, light's going to have a hard time getting to algae that's more in the center of this bioreactor. As a result, I feel like you need to come up with a really clever way to maintain the growth phase of the algae. Even if you were to like use a different kind of setup, so instead of a stir tank, you go with something like a
I think
pool.
that that technical side question is actually like something a little bit out of the scope of what we might be able to answer. I know that there are definitely scalable ways of growing microalgae because it is currently being evaluated for biofuel and other medical applications in other industries. So I definitely think there are solutions for being able to maintain those growth phases, but like I certainly do not have that expertise.
Yeah, me either. I'm just trying to visualize it, right? Because
Yeah.
when I'm thinking about what I
It's
know
really
about
cool to
how,
see. Like I've seen images and videos of some of these like reactors specifically for algae. So it's definitely very different from like the bioreactors we're used to seeing in like pharmaceuticals, because the pharmaceuticals ones are like enclosed all the way around usually, but like the reactors I've seen for microalgae are typically clear all around to allow the light to move throughout. And also, if we are talking about a very similar system to the transwell, which they are like sharing media but not sharing space technically, then that would be very possible to interface these two kinds of reactors together and then just make sure that you are exchanging that media and those nutrients rather than having those interfacing together in the same tank, which is a possibility. And I wouldn't be super surprised to see, but also it definitely makes the system in ways more simple to keep them like barriered against in two different areas.
So this would be
yeah,
like a dual tank type of setup where
Probably
there's
that.
there's an
yeah,
exchange of meat. Okay, yeah, interesting. Yeah.
yeah, I definitely envision it as like a two separate systems interfacing with one another. And I'd just be really curious to see what one of these bioreactors looks like because in the case where you described a transparent outside where light can come in, I still feel as the algae grows, that light can only penetrate so far into the algae before it significantly decreases, and algae in the middle won't get as much light.
I
And
think
then
it's
even if you put
probably.
a light source on the inside, right?
I'm
Even if you
not.
put
I
it,
don't think that's what they do. I think they circle it around with light sources. So very similar to like hydroponic systems, I believe. But once again, I've only like very briefly ever looked into this.
yeah, I've
Did,
also seen like algae ponds or pools where they have like huge vats of this stuff.
I've
But
never
even
seen.
then, I feel like the surface area, right, of the vat is
Hmm.
going to be a limiting factor in the same way that the volume would be in a
And just to
more
jump in here,
conventional looking bioreactor.
Jordan, you made some interesting points about the scale up and the visibility and monitoring. If it becomes more cloudy or obstructed because of the algae, then if you're implementing sensors and stuff like that, there might be interference with debris, which is something I saw with my own research with the plant calluses. If perhaps some debris breaks off in the media exchange or something like that, so it'd have to be very particular to make sure that that doesn't happen. But definitely solvable. They're all solved
Yeah,
Yeah,
cases.
that's
Yeah.
very solvable. Like I
yeah.
work in automation. If you know that those are potential issues, you have like almost either fail safes or have contingencies or sensors around. The question usually is building like the knowledge to be able to ID and then the ability to also fix it.
Yeah, that was also one question I had because I know there's like a lot of like Hamilton sensors that are designed for very specific types of culture. And if it is, to Max and towards point, cloudy or different, then you're going to need either a totally different type of sensor, which is not cheap, or some sort of different type of setup. The two tanks is very interesting. Also, with the light, it does make me think of in the cultured meat industry, there's a company called Prolific Machines, and they're using light in very different ways in unique bioreactor systems, not using algae, I believe. But I think, similar types of ways light at certain times, I think they also adjust the color of the light. I don't think you need to do that here, but they adjust the color of the light. They have different solutions when you do get to much bigger scale tanks, because you can't have, a extremely large tank that's completely made out of glass. At least I don't think. Different ways of funneling the media or even everything, through a chamber that has light, and maybe that can be done here too.
Yeah, it's interesting that you bring up specialized lighting because I think that if you were to actually use this in a commercial sense, you would have to have specialized light. Not all light is created equal. Only certain wavelengths of light are actually useful for photosynthesis. So
And
it
I
wouldn't be
will
as simple as putting a light bulb in front of it. For example, you'd have to have light that's specifically in the photosynthetic range to make sure you're not wasting energy.
And building off of Jordan's comment there about light, my knowledge of microalgae and the fact that they both use and evaluated saltwater, brackish water, and freshwater algae types is that most likely different environments result in these different algae might having different light needs. So that could be a consideration going forward if you decide to use a different species. Maybe if you get really wild here, you have different species that are best suited for different cell types or different cell environments or manufacturing processes. It becomes a question of having that flexibility in order to deal with all these different environments and conditions.
Before we start wrapping up, I do want to kind of go back into kind of like the discussion about powdered media.
Mm.
And maybe you guys can give me an overview of when you would use powdered media in the lab scale, or how they do mix kind of larger amounts of powdered media. Shed any
Jordan
light
can
on that?
definitely. Jordan was the one who did the deep research into how you could powder media. So I'm going to give this to him.
Yeah, pretty much. I feel like most people resort to a powdered media when cost is of utmost importance, which is definitely the case here with this study because that's the whole point in reducing the cost of media for a commercial product. But it's relatively simple when it comes to the base components, right? You have your sugars and everything you can get in powder form, and you just essentially mix it all together in one big batch. But it gets more complicated when you start introducing things like growth factors and things like that. So yeah, I don't think that it would be too different from the way medias are already made related to as it pertains to the topic of this article. I don't think it has much more of an impact if you were to optimize this, right? You might have to include some components. Max can talk about it that might be a little bit more relevant to plants or to algae to help them grow. You want to talk about what might be helpful to include, Max?
I am not an expert on algae particularly, but just knowing different species and even just different species grow differently in different ways in different mediums. With plant cell culture, there's of course the solid state medium as well as the liquid state medium. So adding different components causes them to grow differently, and perhaps I can have some unintentional effects if there is that media exchange, but definitely optimizing those things is something that's super important for scale up.
Great. Are there any other topics that we should dig into?
No, I don't think there are more topics, but I do want to just quickly go over. I think this is pretty good work. I did, there were some things I would like to see in it, such as the differentiation. But most of all, I would like to see them repeat this with meat relevant cells. So maybe bovine satellite cells or QM7s like used
Just send Jordan some microalgae in the mail, guys.
in.
Clearly, it's gonna come from Australia perfectly fine.
Yeah. But yeah, maybe like quail QM7s or maybe a chicken myoblasts, something like that, so we can see a more direct comparison to like how they would grow in this kind of setup and differentiate. I want to see them differentiate.
Cool. Yeah. Well, there's definitely lots of, you said quails. So there's definitely
Yeah.
a lot of cell culturing going on down there.
I don't think they need help.
Thanks everyone for joining. We're going to be adding some show notes like I had mentioned earlier, and of course linking the publication. Before we close out, are there any last insights that anybody would like to mention? I'll direct that question to everybody. Also, the best way for listeners and viewers to contact you, whether it's LinkedIn or email. We can start with Jordan.
Yeah, so my final thoughts, I love the idea of a symbiotic bioreactor, and I really want to see more people start looking into this, and I would really like to see it be applied commercially at large scale. As far as how to best contact me, you can contact me at jordan@jonescellag.com, or you can reach out to me on LinkedIn.
So I really found this article as fascinating as you guys could probably tell. I will send in the link to the article I found on desiccation possibilities for different types of microalgae. And to contact me, I'm best found on LinkedIn at Rebecca Lee. Unfortunately, there's a lot of me, so good luck.
Yeah, I think that this was a really awesome article. I'm super passionate about the industrial bioeconomy, industrial biomanufacturing, and I think that we're going to start seeing some really creative solutions. I'm personally super interested in to see how artificial intelligence and machine learning can help stuff like this. Really excited to see how this plays out. And you can find me best on LinkedIn at Maxwell Levine, and I also have a Twitter at Max Levine. And yeah, that was awesome.
Awesome. Thank you guys. We'll see you on the next episode of Journal Club.